Welcome back, everyone. Ready for another deep dive.
Always.
Today, we're shrinking down, way down, to the level of human cells.
Oh, this is going to be fascinating.
It is, and we've got a college-level textbook excerpt to guide us through the intricacies of cell structure and function.
You know, it's easy to forget just how much is going on in those tiny worlds.
Right, we're talking trillions of cells working together.
It's mind-boggling when you think.
Totally.
about it.
So, before we get into all the details,
Yeah.
our source material starts with this really cool comparison to give us a sense of scale.
Okay.
Imagine like an ostrich egg.
Okay, got it.
Now shrink that down to the size of a human egg cell.
Whoa, that's a big difference.
Huge. And a human egg cell is just barely visible to the naked eye.
Wow.
But even that egg cell is massive compared to most human cells.
Really?
Most of our cells are measured in micrometers.
Micrometers.
It's 1/1,000,000th of a meter.
Okay, so we're talking tiny.
Tiny. Most cells are somewhere between 1 and 100 micrometers.
So you need a microscope to even see them.
Absolutely. Which is why scientists use these incredible tools to explore the cellular world.
Yeah, what kind of microscopes are we talking about?
Well, our source material mentioned three main types.
Yeah.
The light microscope, the LM,
Okay.
the transmission electron microscope, or TEM,
Uh-huh.
and the scanning electron microscope, CM.
So each of these gives us a different view of the cell.
Exactly. The LM is kind of like your basic workhorse microscope.
Okay, for seeing the basic structures.
Right. But when you want to see those really fine details,
You need the big guns.
you need the electron microscopes.
That's the TEM and the SEM.
Exactly. It's like upgrading from a magnifying glass to a super high-powered telescope.
Cool. So what can you see with these electron microscopes?
Well, the TEM lets you see incredibly detailed cross-sections of cells.
Like slicing it open and looking inside?
Yeah. Our source material gives the example of cilia,
Cilia.
those tiny hair-like structures on the surface of some cells.
Right.
With a TEM, you can see the intricate internal structure of cilia.
Wow, that's amazing.
And in the SEM,
yeah.
that gives you a three-dimensional view.
Whoa, like a 3D movie of a cell.
It's pretty close. You can examine the surface of cells and other structures in incredible detail.
That's awesome. It must feel like you're exploring an alien landscape.
Totally.
Yeah.
Okay, so we've got these awesome tools to visualize cells.
Yeah.
But what are the actual building blocks, the fundamental components of these tiny worlds?
Right, good question.
Well, almost every human cell has three core components:
Right.
the plasma membrane,
Uh-huh.
the cytoplasm,
Okay.
and the nucleus.
The nucleus, that's the control center, right?
That's the control center. We'll get to that.
Okay.
But first, let's start with the plasma membrane.
All right, the gatekeeper.
The gatekeeper, exactly. It's
Yeah.
often described as the gatekeeper of the cell.
Controlling what goes in and out.
Right, controlling what goes in and out.
So what's it made of?
Well, it's actually a really remarkable structure. It's made up of what's called a phospholipid bilayer.
A phospholipid bilayer.
And this bilayer, it has these hydrophilic heads.
Hydrophilic, so they like water,
They love water. And then it has hydrophobic tails.
which hate water.
Exactly. They repel water.
So the plasma membrane creates a barrier then,
It does, a barrier between the watery environment inside the cell
right?
and the watery environment outside the cell.
Got it.
And this barrier is what's called selectively permeable.
Selectively permeable, what does that mean?
It means it can regulate what goes in and out.
Ah, so it's not just a solid wall?
Nope, it's very carefully controlled.
So what else is in this phospholipid bilayer?
Well, we've also got cholesterol molecules.
Cholesterol, like what's in our blood?
Yeah, similar. But in the membrane, they act like reinforcing beams.
Okay, so they make it stronger.
Exactly. They strengthen and stabilize the membrane.
Interesting.
And then, of course, there are proteins.
Ah, proteins, those are essential for so many things.
They are.
Mm-hmm.
And in the plasma membrane, they have a bunch of crucial roles.
Like what?
Well, some proteins act as channels.
Channels.
Yeah, they create pathways for specific substances to pass through.
So they're like the gates and the gatekeeper?
Exactly. And other proteins are receptors.
Receptors.
They bind to specific molecules outside the cell,
Okay.
and then they trigger a response inside the cell.
So they're like the communication system.
Exactly. Like the cell's way of talking to the outside world.
I like that analogy.
Now, there are two main types of membrane proteins.
Okay, what are they?
Integral proteins
Uh-huh.
and peripheral proteins.
All right, so what's the difference?
Integral proteins are embedded right in the membrane.
Embedded, okay.
Yeah, they often span the entire width of the membrane.
Wow.
Peripheral proteins are more like they're attached to the surface.
So inside or outside of the membrane.
Right, either the inner or outer surface.
Interesting.
And each type of protein has its own special job.
It's like a well-oiled machine.
It really is. Now, what about these things called glycolipids and glycocalyx?
Glyco? What?
Yeah, they sound a bit complex.
They do?
But think of them as the cell's ID card.
Okay, the ID card.
Glycolipids are lipids, fats,
Right.
with these little carbohydrate groups attached.
Okay.
And they're located on the outside surface of the membrane.
Uh-huh.
And these carbohydrates, along with the carbs attached to the proteins,
Yeah.
they form what's called the glycocalyx.
Glycocalyx, all right.
It's basically a sugary coating on the cell,
A sugary coating.
and it plays a key role in cell-to-cell recognition.
So cells can recognize each other.
Yeah. It's like a secret handshake so they know who's who.
That's pretty cool.
It is. And it's vital for all sorts of processes,
Like what?
like immune responses, tissue formation.
So your immune system uses that to identify your own cells.
Exactly. Imagine if your immune cells couldn't recognize your own cells.
That would be bad.
That would be very bad.
So the plasma membrane is pretty important.
It's crucial. It's so much more than just a simple barrier.
Right. It's dynamic. It's got all these functions.
Right. It's maintaining the integrity of the cell,
Yeah.
and it's facilitating all these interactions with the environment.
And we haven't even gone inside the cell yet.
Nope. There's a whole other world waiting for us in the cytoplasm.
Well, lead the way. I'm ready to see what's happening in there.
Let's go
Okay, so we're like crossing the border now into the cytoplasm.
into the cytoplasm, right?
It's the space between the plasma membrane
Uh-huh.
and the nucleus.
Okay, so if the plasma membrane is like the city wall,
Yeah?
what's the cytoplasm like?
Think of it like a bustling marketplace,
Okay,
crowded with people and goods moving in every direction.
I like that.
First, you've got the cytosol.
Cytosol.
That's the thick fluid that everything kind of floats in,
Okay.
like the air, but more viscous.
More viscous, thicker.
And it's packed with all sorts of stuff.
What kind of stuff?
Ions, nutrients, the building blocks for bigger molecules.
So it's like the cell's transportation system,
Yeah, you could say that.
moving things around, providing the raw materials for building and repair.
Exactly. And within this bustling marketplace,
Yeah?
we find the cell's storage units.
Storage units.
Yeah, they're called inclusions.
Inclusions, okay.
Some cells, like in our skin,
Yeah.
they store pigments like melanin.
Melanin, that's what gives us our skin color.
Right, and it helps protect us from the sun's rays.
Like little sunscreen compartments within the cell.
Exactly.
That's so cool.
And other cells store energy reserves,
Like what?
like glycogen.
Glycogen. Okay.
It can be broken down to provide fuel when needed.
Like tiny fuel tanks within the cell.
Precisely. And then, of course,
Right.
we have the stars of the show, the organelles.
The organelles, okay.
These are like specialized structures. They
Oh, yeah.
carry out specific functions within the cell.
So like they each have their own job to do.
Right. Their own unique role to play.
Okay, so where do we start with these organelles?
Well, let's begin with the membrane-bound marvels.
Membrane-bound marble.
Yeah, the organelles that have their own little compartments.
Oh, okay. Their own little mini worlds.
Exactly. So let's head over to the endoplasmic reticulum,
Endoplasmic reticulum.
or ER for short.
ER. Yeah.
It's a network of interconnected membranes.
A network of membranes.
Yeah, and it winds throughout the cytoplasm.
Wow. So it's like a maze.
Kinda, yeah.
Okay, so what does this ER do?
Well, there are actually two types.
Two types, okay.
Smooth ER
Uh-huh.
and rough ER.
Smooth and rough. What's the difference?
The rough ER is studded with ribosomes.
Ribosomes, those are the protein-making machines, right?
That's right. So the rough ER, it's like the cell's manufacturing hub,
Okay, the factory floor.
churning out proteins.
So those proteins are for what?
Some are destined for export outside the cell,
Okay.
some are inserted into membranes,
Right.
and some are packaged into lysosomes.
Lysosomes, we'll get to those later, right?
Right, those are the cleanup crew.
Okay.
So if the rough ER is the factory floor,
Yeah.
what's the smooth ER doing?
Good question.
The smooth ER has a different set of responsibilities.
Okay, like what?
It's involved in lipid synthesis.
Lipid synthesis, so making fats,
Exactly. Carbohydrate metabolism.
breaking down carbs.
Uh-huh. And detoxification of drugs and alcohol.
What? The cell can detoxify drugs and alcohol.
Well, the smooth ER can.
That's amazing. So, like, if you have a drink,
Oh, yeah.
your smooth ER is working overtime.
You could say that.
That's so cool.
Now, imagine these newly made proteins.
Yeah.
They need to be transported to other parts of the cell.
Okay, how does that happen?
That's where transport vesicles come in.
Transport vesicles, what are those?
They're like tiny little delivery trucks.
Tiny trucks, I like it.
They butt off from the ER,
Uh-huh.
and they shuttle the proteins to the Golgi apparatus.
The Golgi apparatus, that's the one that looks like stacks of pancakes, right?
That's the one. It's made up of these stacked, flattened sacks.
Sacks, okay.
It's called cisternae.
Cisternae, all right.
And it acts as the cell's packaging and sorting center.
So the proteins arrive in their little delivery trucks,
Yep.
and then what?
Think of it like a post office.
Okay.
The Golgi receives the proteins, modifies them, sorts them,
Sorts them, okay.
and packages them for their final destination.
So where do they end up?
Well, they can go to lysosomes.
Lysosomes.
Or
Okay.
secretory vesicles.
Secretory vesicles, those are the ones that release stuff outside the cell, right?
Yeah, that's right.
Old organelles.
Making sure the cell stays clean and tidy.
Exactly.
And they also play a role in defense, right?
That's right. Digesting harmful bacteria that might enter the cell.
So they're like the cell's garbage disposals and security guards all in one.
A very apt description.
It's amazing how these tiny compartments can do so much.
They are truly remarkable.
Are there any other organelles involved in maintaining this internal environment?
Yes. Let's move on to the peroxisomes.
Peroxisomes, okay.
They're like specialized detox units.
Oh, different from the lysosomes.
Yeah, lysosomes are more general purpose.
Okay.
Peroxisomes are really good at breaking down certain harmful substances,
Like what?
like hydrogen peroxide.
Hydrogen peroxide, that's the stuff we use for cuts and scrapes.
Exactly. It's also a byproduct of many cellular processes.
So the cell produces its own hydrogen peroxide.
It does. And peroxisomes use oxygen to neutralize it,
Neutralize it.
protecting the cell from damage.
So the peroxisomes are like the cell's hazmat team.
Very good.
All right, what else? Well, now for the powerhouse of the cell.
The powerhouse, okay.
The organelle responsible for generating most of the cell's energy.
That's the mitochondria, right?
The mitochondria, yes.
Those are the tiny power plants.
Exactly.
What makes them so special?
Well, for one, they have a double membrane,
Double membrane, okay.
and the inner membrane is folded.
Folded, how so?
It's folded into these finger-like projections.
Projections, okay.
It's called cristae.
Cristae, all right.
This increases the surface area.
So more surface area, more energy production.
Precisely.
Clever design.
And here's where it gets even more fascinating.
Okay, I'm intrigued.
Mitochondria have their own DNA,
Wait, what? Their own DNA?
separate from the DNA in the nucleus.
So, like, they have their own little instruction manual.
In a way, yes.
What does that mean?
Well, it suggests a fascinating evolutionary history.
Okay, tell me more.
Scientists believe that mitochondria were once free-living bacteria.
Whoa, bacteria.
Yeah, and they were engulfed by ancient cells.
Engulfed, like eaten.
Kinda, yeah.
And then they just stayed there.
They formed a symbiotic relationship.
Symbiotic, so they both benefit.
Exactly.
That's mind-blowing.
It is.
So these little powerhouses within our cells
Yeah.
were once independent organisms.
It's a remarkable example of how life on Earth has evolved
Through collaboration.
through collaboration and interdependence.
Wow. Okay, so we've covered the membrane-bound organelles.
The ones with their own little compartments.
Right, what about the other stuff in the cytoplasm?
The non-membrane bound stuff.
Yeah, are there any key players there?
Absolutely. Let's start with ribosomes.
Ribosomes, those are the protein synthesis machines.
That's right. And you'll recall we saw them on the rough ER.
Right, making proteins for export.
Uh-huh.
Are there other types of ribosomes?
Yes. We also have free ribosomes.
Free ribosomes, what do they do?
They float in the cytosol,
Okay,
and they manufacture proteins that are needed within the cell itself.
so it's all about location, location, location.
Yeah.
Ribosomes on the rough ER, protein shipped out,
Yeah.
free ribosomes, proteins for the cell's own use.
Perfect.
Okay, what else?
Now let's talk about the cytoskeleton.
Cytoskeleton,
It's the cell's internal framework.
internal framework, so like the scaffolding,
Exactly. It's a network of protein filaments.
filaments.
It provides structural support,
Uh-huh.
helps maintain cell shape,
Okay.
and even enables movement.
Movement, wow, the cell can move.
Well, some cells can, yeah.
Okay, so the cytoskeleton is like the bones of the cell.
You could say that, yeah.
All right, what are these filaments made of?
There are three types.
Three types, okay.
Microfilaments, intermediate filaments,
That's
and
it.
microtubules.
Microtubules. All right, let's break those down, starting with microfilaments. What do
Uh-huh.
those do?
Microfilaments are the thinnest of the three.
Okay.
They're involved in cell shape changes,
The cell shape changes. Interesting.
muscle contraction,
Muscle contraction. Wow.
and cell division.
So they're like tiny muscle fibers,
You could say that, yeah.
providing the force for these actions.
Exactly.
Okay, what about intermediate filaments?
Intermediate filaments are more like structural beams.
Okay, providing support.
Right. And they stabilize cell junctions.
Cell junctions, those are like the connections between cells, right?
Exactly.
So it's help hold everything together,
They are especially important in tissues that need to withstand stress.
like muscles and skin.
Exactly.
All right, and lastly, we have microtubules.
Microtubules, these are the largest of the three filaments.
Okay.
And they're like cellular highways.
They're highway.
Yeah, they form tracks
Okay.
that guide the movement of organelles and vesicles.
So those little delivery trucks we talked about earlier,
Exactly.
they travel along these microtubule highways.
Precisely.
Wow, that's so cool.
And microtubules also play a crucial role during cell division.
Cell division, how so?
They form the spindle fibers,
Spindle fibers.
which
Wow.
separate the chromosomes.
So microtubules are involved in a lot of important stuff.
They are.
Okay, what else is going on in this cytoplasm?
Well, have you ever wondered how some cells move around?
Good question.
Or how they move fluids and particles across their surfaces?
Well, I know sperm cells can swim,
Right.
and I've heard about cilia in our respiratory system
Cilia.
that helps sweep away mucus.
Those are great
Wait,
examples.
look, okay.
Both cilia and flagella.
Cilia and flagella.
They're hair-like projections.
And projections, okay.
That extend from the cell surface.
Okay.
And they're powered by microtubules.
Microtubules, again, they're busy.
They are.
So cilia and flagella are like tiny oars,
Exactly.
propelling cells through fluids or moving substances.
Precisely.
So cilia are shorter and more numerous,
Right. Like hair.
found on cells' lining surfaces,
Like the respiratory tract.
sweeping away mucus and debris.
Exactly.
Flagella are longer, usually just one,
Yeah, like a tail.
and they propel entire cells.
Like sperm cells.
Okay, that makes sense.
It's fascinating how these tiny structures can generate movement.
It really is.
Is there anything else we should know about these non-membrane bound components?
Anything else in the cytoplasm?
Yes. One more structure. Microvilli.
Microvilli. Okay.
These are tiny finger-like projections,
Projections. Oh,
and they increase the surface area of the plasma membrane.
the plasma membrane. Okay.
Why is that important?
Good question.
They're particularly important in cells that are involved in absorption,
Absorption.
like the cells lining the small intestine.
Right. Those are absorbing nutrients.
Exactly.
So by increasing the surface area,
Yeah,
they can absorb more nutrients.
precisely.
It's all about efficiency.
Exactly.
Wow. Okay. So we've explored this bustling marketplace that is the cytoplasm.
We have
From the transport systems
Mhm.
to the storage units,
the recycling centers,
the power plants.
the intricate framework.
It's incredible.
It really is.
So I think it's time we make our way to the nucleus,
The control center.
the heart of the cell.
Let's go.
Okay, we've made it. The nucleus.
The heart of the cell,
The control center.
where all the genetic blueprints are kept safe.
It's like the cell's library,
Right. A perfect analogy.
containing all the knowledge to create and sustain life.
And just like a library, the nucleus has its own set of specialized structures.
Okay, so what are these structures? What keeps those precious blueprints safe?
Well, first we have the nuclear envelope.
Nuclear envelope,
It's a double membrane that surrounds the nucleus.
like a protective barrier.
Exactly. And it has pores that control what comes in and out.
So it's like the security checkpoint at the library.
Precisely. Only authorized molecules can enter or exit.
Love it.
And within the secure vault,
Yeah,
we find the nucleolus,
the nucleolus.
another distinct structure within the nucleus.
So what's the nucleolus doing?
The nucleolus is like a ribosome factory.
A ribosome factory.
It's where those protein-making machines are assembled.
Ribosomes, right?
Yes. Before being shipped out to the cytoplasm
So the nucleolus is churning out the tools the cell needs.
to build and repair itself.
Fascinating.
Oh.
Okay, so now let's talk about the genetic material itself,
The DNA.
the DNA.
Our source material mentions DNA, chromatin, and chromosomes.
Right, and I'm always a bit confused about the difference between those.
It's all about levels of organization.
Okay,
Think of DNA as the raw material, the actual blueprint.
the blueprint. Okay, got it.
Chromatin is DNA packaged with proteins.
Packaged.
Imagine the blueprint, but rolled up neatly.
Okay, so it's more organized.
Exactly. This packaging helps condense the DNA.
So it fits in the nucleus.
Right, and it regulates which genes are accessible for protein synthesis.
So it's not just storage, it's also control.
Precisely. And then we have chromosomes.
chromosomes.
Those are the highly condensed form of chromatin.
High condensed.
Visible only during cell division.
Okay, I think I'm starting to get it.
It's like the blueprint organized and ready for distribution.
So when the cell is not dividing,
Yeah.
the DNA is spread out as chromatin.
Right, so it can be read.
But when it's ready to divide,
Then the chromatin condenses into these compact chromosomes.
okay, that makes sense.
Yeah.
And this brings us to the cell cycle.
The cell cycle, yes.
Our source material talks about interphase and the mitotic phase.
The two main phases of the cell cycle.
Right, so let's break those down,
Wow.
starting with interphase.
All right.
Is that when the cell is just kind of resting?
Not at all. Interphase is a very busy time for the cell.
Oh, really?
It's the period between cell divisions,
Okay.
and it's divided into three phases, G1, S, and G2.
G1, S, G2. All right, what are those all about?
G1 is a period of growth.
Growth, okay.
The cell increases in size, produces new organelles, synthesizes proteins.
So it's stocking up on supplies.
Exactly. Preparing for DNA replication.
And then comes the S phase.
S for synthesis.
Synthesis. So that's when the DNA replicates,
You got it. Each DNA molecule makes an exact copy of itself.
making sure each daughter cell gets a full set of instructions.
Precisely.
Okay. And what about G2?
G2 is another growth phase.
More growth, okay.
But this time, it's about final preparations for cell division.
Like the final dress rehearsal.
Exactly. The cell checks its DNA for errors.
Make sure everything is in order.
Make sure it has enough organelles,
Hmm.
synthesizes the enzymes needed for mitosis.
So after G2, the cell is prepped and ready for mitosis.
Ready for the big event.
So mitosis, that's the actual process of nuclear division, right?
Exactly. And it's divided into four phases: prophase, metaphase, anaphase, and telophase.
Okay, let's go through those, starting with prophase.
All right, during prophase, the chromatin condenses into those visible chromosomes.
The ones we talked about earlier.
Exactly. The nuclear envelope breaks down and the mitotic spindle begins to form.
The mitotic spindle, what's that?
It's a structure made of microtubules.
Microtubules, the cellular highways.
That's right. The spindle fibers attach to the chromosomes
Yep.
and help to orchestrate their movement during mitosis.
So prophase is all about getting organized for the split.
Exactly. Then comes metaphase.
Metaphase.
Metaphase is all about alignment.
Alignment, okay.
The chromosomes line up neatly at the center of the cell.
Like they're all lined up for inspection,
Precisely. Attached to those spindle fibers.
ready to be divided.
Exactly. And then anaphase.
Anaphase.
Things start to get exciting here.
Okay, what happened?
The spindle fibers shorten,
Shorten, okay.
pulling the sister chromatids apart.
Sister chromatids, those are what make up each chromosome, right?
Exactly.
So they get pulled apart.
Toward opposite poles of the cell.
So each chromatid becomes a separate chromosome.
Precisely.
And finally, telophase.
Telophase.
What happens in this last stage?
During telophase, the chromosomes reach the poles.
Okay.
The nuclear envelope reforms around each set of chromosomes.
So we have two nuclei now.
Exactly. And the chromosomes unwind back into chromatin.
Back to where we started, but with two nuclei.
Precisely.
But what about the rest of the cell?
That's where cytokinesis comes in.
Cytokinesis, okay.
The division of the cytoplasm.
So the actual cell splits in two.
Exactly. It usually happens at the same time as telophase.
How does that work?
A contractile ring of protein filaments.
Protein filaments, okay.
It pinches the cell into, like, tightening a belt.
And then two daughter cells.
Two identical daughter cells.
It's incredible how this all happens so precisely.
It's like a perfectly choreographed dance.
And it's happening in trillions of cells right now.
All throughout your body.
It's amazing to think about.
It really is.
Well, this deep dive has certainly been eye-opening for me.
It's fascinating stuff.
I've learned so much about the complexity and wonder
Yeah.
of this microscopic world.
It's a reminder that even though we can't see them,
Cells are constantly working to keep our bodies functioning.
absolutely,
So we encourage you to keep exploring this amazing world.
there's always more to learn.
Thanks for joining us on this deep dive.